Hello, and thank you to the organizers for inviting me to give this talk about the Visium technology that can be used to conduct spatial transcriptomic on tissue sections. But first, I want to go over some of the features of Visium technology. And first off is the sensitivity of Visium technology is that you can get thousands of genes detected per spot, and there are about 5,000 spots per capture area for resolution, each spot measuring about 55 micron diameter. And this can fit about 1 to 10 cells per each spot, depending on your tissue and the size of your cells. Additionally, each spot is able to capture all of the mRNA, so you can get the entire transcriptome and not specifically targeting any specific mRNAs. And lastly, this workflow from getting your tissue section onto the slide to library preparation and ready for sequencing can take about 1 day time. And just looking briefly at the Visium slide here, you can see in each slide there are about 4 capture areas per slide. And each of these capture slides have those 5,000 spots. The red spots around the larger opening is allowed for alignment during the analysis portion and does not actually capture any mRNAs, but anything covered by the black spots will be sequenced during the Visium process. And lastly, there is another Visium slide available that has 8 capture areas that's just for optimization on the permeabilization step, which is during the workflow. That step only needs to be optimized once for each tissue type. Once you know how long that step's going to take, you use the gene expression slide, which has the 4 capture areas. The entire workflow here is laid out. First, you'll do your tissue preparation and get your tissue blocks. You'll slice them from about 5 to 10 microns thick. That is then placed onto the tissue prep slide. Next, they are imaged, which allows you to see your tissue image. They are then permeabilized, which will allow your mRNAs to come out of the cells and drop into the spots underneath. Those spots then align to the probes that are under there. Each probe also includes a sequence tag. That sequence tag is then used afterwards during the alignment process and to deconvolute each mRNA to the initial spot on the spatial XY plane where that came from. And that's kind of how you're going to connect the spatial tissue section to the gene expression. To finish up library prep, those probes that have aligned to the mRNAs are then used to create the cDNA. Those are then sent to your sequencer. Eventually, you'll get back your FASTQ files that you can align to your genome, as well as do your data visualization. For this data visualization and alignment step, 10x does provide 2 software. One is Space Ranger, which conducts your alignment and some of your initial analysis. You can visualize it using their Loop browser, which is their second software. You can also take the aligned reads from Space Ranger and send it into R or other analysis platforms to analyze using other packages, including Seurat, if you want to do anything beyond what the Loop browser is able to. And so I want to highlight 2 projects that are ongoing in our lab using the Visium technology. First is looking at orchiectomy and the mouse prostate. And second is looking at human prostate. So the first one looking at mouse prostate orchiectomies. So in here we took 15-week-old black 6 mice. We did a sham surgery on 2 of them, and 2 of them we performed the orchiectomy. And after 15 days, we dissected out the prostates, removed seminal vesicles, and embedded into the blocks. These blocks are then sliced, and we initially did H&E staining to figure out where in the blocks we wanted to actually do the Visium sequencing. Because this is the first time we used mouse prostate, we did do the tissue optimization for the permeabilization time. We just had to do that once. So if we want to repeat this experiment, we don't need to do the optimization again. Once we knew that time frame, we then took the sections to be analyzed into that gene expression slide with the 4 capture areas. We imaged it, parameterization, all of the steps for library construction, and sent for sequencing. And as we were kind of waiting for that sequencing, we were able to look at that mouse tissue sections and identify some of the prostate lobes. Up here on the right is just kind of a legend there. So the first set of tissues up on the top is the initial H&E sections that we used to identify where in the tissue blocks we were going to pull out for Visium. On the bottom is the actual Visium sections that were sequenced. As you can see, this is the kind of colors and that you'll get using the Visium. In here, all of the circled in or yellow is the dorsal lobe. Red circles is what we believe is the ventral lobe. Blue is more your anterior lobe. And we did find pieces of the urethra circled in green in some of the sections up top. And when we looked at the initial H&E sections, we found in good pathology to look at the different lobes. And we're finding again some of those lobes in the sections that were sequenced using Visium. So we know that we're going to be able to analyze some of these lobes, especially the anterior lobes that we're finding in all 4 sections. So once we sequence them, and here you can see the UMAP of all spots across all 4 sections that had tissue overlay on them. And we were able to break it down using gene expression into a lot of different lobes, including some sections that we weren't expecting, such as some neuronal-like spots that were found there. And on the right here, you'll have for one of the sham mouse, the initial pathology that we circled, looking at finding all the different lobes. And in the middle is actually each spot colored by the same color as the clusters found in the UMAP. And you can see they're really breaking down across there. We're seeing some of the gene expressions in the middle where it's orange and red. That is very anterior lobe-like that we were making the initial pathology in the Visium initially. Additionally, we were able to further subsect the dorsal lobe that you wouldn't have been able to do if you had just sliced it out and sequenced it completely, or even done in some of the single cell without some of the spatial information, is in the surgical mice and orchiectomy mice. We were able to find the dorsal lobe. All of the spots are very similar in gene expression. However, in the sham mice, the dorsal lobe, we were able to further subsection into spots that showed a lot of the usual gene markers for dorsal lobe, but also some of these spots showed a lot of muscle cell expression. So if we were just to section these out, we would have missed some of these expression, knowing that these muscle cells were linked with the dorsal lobe, so we can look at them specifically. We can also look at them in conjunction with the orchiectomy mice, or just look at the dorsal lobe spots that show high levels of the expected gene expression to look at what other differences are in there following the surgery. I'm just briefly going over the second study we used using Visium technology of the human prostate. And in here, we collected human prostate cancer tissue, we processed and prepared it and sent it through the Visium pipeline. And we initially looked at the differences based on a microenvironment that we saw in the pathology, though further study and exploration can be used on these sections. And so briefly what we did is we looked, as you can see here in kind of panel A, is first the initial slide that was put onto there, and looking at some of the pathology, we were able to see some high microenvironment cells that were invading into there, or low microenvironment, as you can see on the bottom. We also overlaid it using the colors that were identified in the clusters using the UMAP gene expression of all of the spots, which is seen in kind of panel B. It identified generally 18 clusters. And in C, the UMAP really separated out those tissues that we saw were kind of high microenvironment and low microenvironment, suggesting there are some underlying genetic differences that in an overview you can see in panel D. There are some gene differences into here. So further exploration will need to be looked into to see if there's any specific gene subtypes that we're finding in there, or any specific genes that are driving these changes. So just kind of an overview of what Visium offers. It's one way to analyze your gene expression profile in your spatial context. And these libraries can be prepared from tissue sections in about 1 day. Sequencing and data analysis can be performed using Space Ranger and Loop Browser, though if you have a favorite package you want to use in R or other applications, you can. That's very much kind of open. And Visium can be used to find genes of interest without having any prior knowledge of your tissue that's being analyzed. And with that, I'd like to thank the Keller Lab, especially Greg Shelley and June Escara-Wilk. They did a lot of the work on the 2 projects that I talked about in here. And with that, I'd like to thank again, the organizers for inviting me to give this talk. And if there's any questions, I'm happy to answer them. Thank you.